I'd like to welcome Jeremie Piña from NIH. He and his colleagues have a paper in the JDR special issue on imaging that describes the use of multi-omics and spatial imaging in deciphering the pathogenesis of cleft palate in a mouse model. Hello, Jeremie. In broad terms, please describe the different types of single-cell analysis that you integrated in your paper.
Sure. Thank you, Dana. So we really integrated three main types of single-cell analysis. One was the RNA at the gene expression level that was coupled with the epigenetic information or the chromatin accessibility information. And then finally, that was integrated with the spatial arrangement of gene expression using a highly multiplex spatial RNA sequencing platform.
That sounds very interesting. What did you learn from the imaging aspect of this?
As a developmental biologist, we learned, really importantly, where these genes are located in time and in space, which we feel is perhaps the most meaningful information, to know exactly where those RNA molecules are located within the tissue of interest. It's a higher level of insight than the single cell RNA sequencing alone.
I can understand that. Did the different approaches provide consistent results, or in some cases, were there road bumps in which integrating the data was problematic?
That's a constant question on our minds, right? We always want to verify our results between these different platforms and technologies. What I can say is that with every step we took, we got a little bit deeper into the data sets, and we realized there's a lot to analyze when you start to sequence the genome, the transcriptome of all of these cells. So I would say that the spatial information really gave us the deepest understanding, whereas the single cell RNA sequencing was more of a bird's eye view of what was going on in the cells in general. We could then localize it directly to the palate along the edges that we needed to understand.
Yes, that's a great answer. What was the most challenging aspect of the spatial imaging?
I really think the tissue quality is probably the most difficult for many labs to optimize when they are running these experiments. Fortunately, our lab is heavily histology based, so we were well groomed to kind of make this transition into the spatial sequencing field, because we do so many very basic, histology imaging and analysis in our lab. So I think that for labs to transition into this field, they should really be focused on preserving and processing their tissues accurately, and then, of course, orienting them on the slide properly. That can really make all the difference.
What is a key take-home message regarding the insight provided by bioimaging and multi-omics in the study of the cleft palate?
For us, the key insight that we gleaned was not only a key signaling pathway that we were familiar with. The Wnt signaling pathway is involved in this complex, palate development process, but a specific biomarker of that Wnt signaling pathway was isolated and identified right at the edge of the cleft palate that is failing to form. So there could be diagnostic and therapeutic implications of this biomarker that we'd like to explore further.
And is there any evidence that this occurs in humans?
There is. With the advent of these genome-wide sequencing studies that are performed across the world, we've actually seen some relevant hits in several of these biomarkers that we identified in our mouse model that overlap with the human genome in patients that are affected with craniofacial malformations. So we're hopeful that there could be some translational relevance to these findings.
Mm-hmm. And I'd like to end with a more personal question, and that is, are there a few things you have really enjoyed as an investigator in dental research?
Oh, I love that question, because this is an exciting time to be an early stage investigator in this field. I think what I love the most is somewhat somewhat ironic because I feel like our field is lacking in, many individuals who are really passionate about this field of research. And I kind of love being on the forefront of, creating new questions that need to be answered and looking into things at a deeper level while also, pursuing my clinical training in dentistry. I feel that this is a unique time to bring science and technology forward as a dental researcher and hopefully come up with some new solutions for the future.
Mm-hmm. And what are your clinical studies on, or your clinical experience?
Sure. Yeah. Well, currently I'm completing my DDS degree at the University of Maryland School of Dentistry, and I hope to apply for residency in oral and maxillofacial surgery. And the dream job would be, taking care of these patients that have cleft anomalies and craniofacial disorders. That's really where my heart is clinically and with research. So I'd like to try to couple these two in the future.
Yeah, it seems like a great integration, not only of scientific platforms, but of your personal clinical and scientific goals.
I hope so. That's really my hope and my dream for the future.
All right, Jeremy, thanks so much. This has been very informative, and it's been a pleasure to meet you.
Likewise, Dana. Thank you so much.